Good morning and good afternoon. I'm Christina Bowens, Global Market Development Manager for Digital PCR at Thermo Fisher Scientific. Today, we're joined by experts who will discuss the state of liquid biopsy research and the ways that cutting-edge genomic technologies like digital PCR and next-generation sequencing can impact clinical cancer research across different cancer types and applications. Throughout the course of this discussion, please enter your questions into the chat box provided and stay tuned for an after-event summary where we'll post the most frequently asked questions and the responses from our panelists on today's discussion. So to get started, let's introduce our panel. First, we have Dr. KJ Kao, who is the head of the Department of Molecular Medicine and Research at the Ku Foundation Sun Yat-sen Cancer Center in Taipei, Taiwan. He obtained his medical degree from the National University of Taiwan and his PhD from Duke University. He was a professor in the Department of Pathology, Laboratory Medicine, and Immunology at the University of Florida. His research focuses on drug target discovery and the development of precision medicine approaches for cancer screening, diagnosis, personalized treatment, and disease activity monitoring. Dr. Claude Pelowitz is the head of research of the Belfer Center for Applied Cancer Science at the Dana-Farber Cancer Institute in Boston, Massachusetts, where he uses his industry experience, government institution experience, and academia experience to translate innovative and novel discoveries into clinical trials at the Dana-Farber Cancer Institute. Dr. Pelowitz has invented and patented technologies in the fields of precision immunotherapy, molecular diagnostics, liquid biopsies, and proteomics. He earned his PhD with honors from Georgetown University and his Bachelor of Science degree from Baldwin Wallace College, and has co-authored over 125 publications. Dr. Cristina Bilbao is a senior researcher in molecular biology, working in hematology service in Hospital Universitario de Gran Canaria. She obtained her degree in biochemistry from the University of Oviedo, Spain, and developed her PhD at the Burnham Institute, La Jolla, California. She teaches cell biology at the University of Las Palmas de Gran Canaria, and her research focuses on the molecular basis of hematological disorders. Dr. Luca Quagliata is serving as Vice President and Global Head of Medical Affairs for the Clinical Sequencing Oncology Division at Thermo Fisher Scientific. Before joining Thermo Fisher, Dr. Quagliata worked as Senior Director for the R&D unit and as the team leader of the Molecular Diagnostics Division at the Institute of Medical Genetics and Pathology, University Hospital Basel, Switzerland. Dr. Quagliata is trained in medical biotechnology, holds a PhD in vascular medicine, and is a board-certified specialist in medical affairs. Dr. Quagliata's team was among the first in Europe to introduce NGS and liquid biopsy testing for routine diagnostics of solid tumors. And finally, Dr. Paul Hung joined Thermo Fisher Scientific in 2021 through the acquisition of Combinati, where he was founder and CEO from 2016 to 2021. Prior to Combinati, he was the Senior R&D Manager at MilliporeSigma, a division of Merck, from 2012 to 2016, managing microfluidic cell analysis product development. Dr. Hung co-founded CellASIC Corporation in 2006 and served as its Chief Technology Officer until it was acquired by Merck in 2012. Dr. Hung earned his Bachelor's of Science in Electrical Engineering from National Taiwan University in 1997 and his PhD in Electrical Engineering and Computer Science from the University of California, Berkeley in 2005. Good morning and good afternoon. I'm Christina Bowens, Global Market Development Manager for Digital PCR at Thermo Fisher Scientific, and today we're joined by experts who will discuss the state of liquid biopsy research and the ways that cutting-edge genomic technologies like digital PCR and next-generation sequencing can impact clinical cancer research across different cancer types and applications. Throughout the course of the discussion, please enter your questions into the chat box provided, and stay tuned for an after-event summary where we'll post the most frequently asked questions and the responses from our panelists on today's discussion. So to get started, if you're tuning into this virtual event, it'll be no surprise that liquid biopsy is an emerging area of clinical cancer research for many different cancer types. Liquid biopsies, which are minimally invasive, enable researchers to collect and study tumor biomarkers like circulating cell-free DNA more routinely, all from a simple blood draw. In the field and across cancer types, we know that there's a wealth of ongoing research devoted to using liquid biopsies as a biomarker-based approach for early detection, for monitoring disease progression, and potential recurrence during and after treatment, and generally to enhance overall understanding of the mechanisms behind disease development and progression. So my first question to all of our panelists is, can you please briefly describe each of your primary research goals and focus areas? And can you explain what you feel the predominant clinical utility of leveraging liquid biopsies is for clinical cancer management? And so I'll pass off to Dr. Palowitz to start.
Thank you so much, Christina. So my research, so I'm head of the Balfour Center for Applied Cancer Sciences. And as part, we have an advanced translational laboratory, the TRL, which focuses specifically on blood-based biomarkers. And as part of that, you have CTCs, cfDNAs, and different modalities of anything shedded into the bloodstream. In the cfDNA, ctDNA space, I focus currently on making tests faster and cheaper for the patient. So my laboratory is uniquely positioned right between the hospital and right between pathology who actually will utilize the tests. So right now it's mostly on the research front, trying to develop assays to be used in the pathology laboratory, and hopefully we can actually get to a point where, what I always say, I want a 20-minute test for $20. So that's my vision. To your second question, which I think is most importantly, so where do we use liquid biopsies? I think we need to sort of like have a heart-to-heart here, is that liquid biopsies are most useful right now in the clinical utility space, in spaces where solid biopsies are needed or need to be supplemented. So this is very late on in the clinical management space. So this is non-invasive genotyping as demonstrated by EGFR mutations like EGFR exon deletion 19, EGFR-LA5R, which used to be activating mutations for first and second generation EGFR TKIs. Then over the last 6-7 years, we sort of like developed assays that actually can predict resistance. So that is the most common space right now where advanced technologies are being used. You can have focused assays such as digital PCR, or you can have broad assays as very deep NGS assays. Now, currently, there's a lot of efforts ongoing to monitor MRD space, early diagnostics, or prognostic of diseases. And they're at different spaces right now. So we sort of move back a little bit more. A lot of researchers currently right now need to understand MRD, minimal residual disease, using a liquid biopsy test and using that sort of information to either intensify or de-intensify treatment options. It's promising. I think it's a little bit early to see if there's clinical development. And then even earlier, we have obviously the holy grail of any kind of liquid biopsy, which is early diagnosis using a test. So those are the earliest development. But the way what I like people to think about it is technology revolutionizes biology and biology drives technology. And I think that's where liquid biopsy is right now. And I would like to get Dr. Kaur's opinion and would like Dr. Kaur to introduce himself as well.
Okay, yeah, thank you for Christina's questions. And our interest in the use of liquid biopsy is to monitor treatment response in cancer patients and detect early recurrence after treatment and detect emergence of specific drug-resistant mutation to kinase inhibitors. And we believe if we can do that, we can optimize the care of patients and achieve better clinical outcomes to patients. And these are our primary goals. And in Taiwan, lung cancer is the number 3 cancer in Taiwan. And unlike patients in most other countries in Eastern Asia, the lung cancer with EGR mutation happens in high percentage of patients. In our hospital, almost 60 to 70% of our lung cancer patients carry EGFR mutations. So we are very interested in using liquid biopsy and to optimize the care to these patients. So, and eventually we would like to utilize this technology to different solid tumors and expand its use. So we are at the same time trying to use the NGS to identify mutation biomarkers in patients, then followed with digital PCR that has high sensitivity and also can be quantitatively applied. And so this is what we are doing. I think this is along the line of many other investigators in this field. That's all. That's what we have been doing.
Fantastic. Now, I know, Dr. Bilbao, you have experience and expertise in hemato-oncology, so I'm really interested to understand more about your primary research goals and where you see the predominant clinical utility of liquid biopsy fitting in the overall cancer research space, and then maybe how you see that fitting more with your line of work.
Hello. Well, I think the hematology is a field where molecular and cytogenetics analysis have been routinely carried out in the last 20 years already for diagnosis, prognosis, and follow-up of myeloid and lymphoid malignancies, especially for myeloid malignancies. And accordingly, the incorporation of new technologies have been occurring. NGS, for instance, which is nowadays mandatory for acute myeloid leukemia diagnosis. And with regard to the clinical advances, target therapies have also been recently developed against the specific mutations in genes such FLT3, IDH1 and 2 in acute myeloid leukemia. Therefore, to search for this mutation and monitor, it is also important under these target therapies. And finally, recent research has shown the importance of the liquid biopsy and the diagnosis and follow-up of minimal residual disease in lymphoma. So I think we have a lot of work to do at this regard.
Absolutely. Thanks very much. So knowing that the fraction of circulating tumor molecules that we're looking at or these analytes that we're looking from blood-based liquid biopsies is often very rare, we can turn to the technologies that are most commonly used to perform the actual analysis portion. So we know digital PCR has pinpoint precision and accuracy for variants that are already known, and NGS really excels with comprehensive screening panels. And these are really the predominant technologies because their ability to detect extremely rare molecules in very small amounts of sample is very high. So my next question to all 5 of our panelists is, where do you think that these genomic technologies like digital PCR and NGS have the biggest impact in human healthcare? And what do you really believe are the top drivers of usage for these genomic technologies across these applications? And I'll start with Dr. Cao.
The advantage of NGS is comprehensive coverage of tumor mutation detection. And the advantage of digital PCR is high sensitivity and quantitative nature. So it's suitable for longitudinal monitoring of the disease. So to make these 2 technologies become routine clinical tests, we have to show proven clinical benefits and affordability and reduced technical complexity for the test. And ideally, digital PCR for the detection and quantification of ctDNA with targeted mutation I believe needs to be more automated. And affordability is an important issue for patients in Taiwan because we have comprehensive national health insurance. Nevertheless, the program currently doesn't cover NGS test and liquid biopsy test due to their relatively high cost. So most patients are unwilling to pay for this test out of their pockets. So the national insurance program will cover this test when the test become more clinically proven, clinically cost effective. So those, I think, are the key factors that would determine the eventual usage of these genomic technologies in the clinical area.
So I'll keep the floor open if Dr. Palowitz or Dr. Bilbao, you have any comments to that.
Well, I think those are really good comments, especially the affordability. If you look at the technology, one could see that NGS could actually be used as early on to actually identify the mutation of interest because it's broad, and then you use actually digital PCR tests to monitor. I think one one issue that is usually not spoken about is that liquid biopsies are actually difficult. It's not easy to really translate this right now into the community setting. You get blood, it needs to be processed appropriately. Do you use a extract tube? Do you use a EDTA tube? How do you get it to the laboratory? Then you spin it down, then you do a second spin, then you actually isolate the cfDNA, then you have your assays. So any step or any sort of technology that actually reduces the complexity, such as Dr. Kao said, like automation, will be a major driver for really applicating. So right now, if you think about 2023, most tests are send-out tests. I mean, the technology is right now in distributed service models with major players where the tests get sent out. Now, that is not scalable. That is really not scalable if you think about using it routinely in the hospital setting. So where the technology is and where I think it should be moved is really to simplify the steps and to make assays more routine. We need to get to the point where a liquid biopsy test is as easily done as a complete blood work count test. And I think the field is moving there. Again, which I think the technology is moving there as well, like such as with the absolute Q. I think that's very helpful. But I think we need to sort of really, it's the beauty of a liquid biopsy test is it's so tangible. A patient can understand it. They really, like you take a blood test, you can understand something about your cancer, and they like to see changes. They like to see that the target treatment works. But we really need to get to the point that we really can draw the blood, get the results, and really engage the patient on the same day.
Absolutely, that's very valuable. And Dr. Bilbao, how do you feel? What are the main drivers of digital PCR and NGS usage across your research areas and applications?
I think NGS, we all know that scans part of the genome that is related to a specific pathology in a single experiment, or even the genome or the exome, right? So that's a great advantage. And on the other hand, the digital PCR allows the direct absolute identification and quantification. For instance, in terms of rearrangement of fusion genes, it's ideal to monitor if you have enough sensitivity that you do with the digital PCR to monitor the disease, right? And also the digital PCR to identify mutations that are in a low burden in a specific tissue, but it is important to identify it in order to diagnose a specific pathology, for instance, or to monitor point mutations that are being followed because you have a therapy directed to them. So I think those are the main utilities in my field.
Absolutely. So turning to our Thermo Fisher panelists, Dr. Hung and Dr. Quagliata, I wanted to get your thoughts and opinions on these two technologies and really to discuss, what's the real difference between platforms, between panels, and the advantages of each. So I'll start with Dr. Hung.
All right, thank you, Christina. And thanks again for the panelists offering us from the near patient's view, from a clinician or clinical researchers or treating patients. So my view as a Thermo Fisher, as a technology developer, is to try to understand these feedback from the clinical researchers, and then try to see how we can improve the patient care at the end of the day. So it sounds like the overall process in the patient's journey start with all the way from early diagnosis to treatment to monitoring, as mentioned by and for different cancer types. And like Dr. Pavla said, it would be great to just know from the blood test, more about what's happening in your body. We do blood tests often. I think everybody have a health check, you do blood draw, but now it's just like, how can we see what else is in the blood? There are typical common biomarkers, but oncology biomarkers wise, digital PCR from our view is about the already mentioned by the panelists, is ability to go deep, higher sensitivity, and something specific. I'll say it's more like a spear fishing. You know what you're looking for, you want to do it very precisely, accurately, reproducibly, then digital PCR will be if that's a question you're going to answer, and digital PCR may offer you the best technology to do that. However, if you don't know what you're looking for in the ocean, then my view is NGS more like it's net fishing. You're going to cast a net, you're going to catch everything, and you're going to be able to see what you're going to get. So our view is the technologies are complementary to each other. It really depending on the biological question you try to answer and in which part of the patient's journey. And then we're fortunately in our case, we have a suite of genomic tools available at Thermo Fisher, so that we believe by working with these clinicians and researchers, we're able to bring tests to be more distributed near patients, and then also, of course, cheaper, faster, and better. That would be my view.
Yeah, fantastic. And that actually turns really nicely into Dr. Quagliata. So would you be able to similarly describe the advantages of NGS and how it differs in terms of the applications from your perspective? And really, what is the environment and resources like for developing oncology diagnostic strategies involving the identification and analysis of circulating tumor events?
Sure. First of all, thank you very much for an invitation, Christina. Let me thank all the participants in here. Already an extremely interesting discussion. I'm going to state the obvious before I start my description, and that is liquid biopsy probably is one of the most promising tools to reshape the way medicine is practiced in the next years. It has been a great promise. I think it's now time to finally turn it into a reality in the clinical space. These are, of course, already used today. There are a number of very solid applications. We heard some of them, especially around EGFR, tyrosine kinase inhibitors, and resistance, but there is much more we can do. And at Thermo Fisher, we are fully committed to continue to develop end-to-end solution with the aim to unleash the full potential of liquid biopsy and to enable our customers and collaborators to continue to develop new solutions for liquid biopsy from the research and down the road to the patient. So having said that, now focusing for a moment on NGS and the ability of NGS to detect multiple biomarkers at the same time, when it comes to Thermo Fisher solution, we today offer a number of different assays, which are all characterized by the ability of detecting both DNA and RNA alterations at the same time. That is also a big challenge in liquid biopsy. We heard about that, as beautiful as it is not easy. And especially when it comes to RNA, for example, RNA fusions in liquid biopsy, that is still a very much open field, which needs to be further solidified. We are there with our solution with the aim of making this possible. So we do have panels that are specifically made for lung cancer, colon, breast, but we also have pan-cancer solution. And again, no matter which kind of panel you're using, all our panel for liquid biopsy are characterized by having extremely low level of LOD, but also taking advantage of our amplicon-based technology, we can use very limited starting input material. But I really want to echo what my colleague Paul just said. It's not just about the technology. It's about using the right technology at the right time for the right patient. And so depending where you are in the journey, as we just said, you might want to be using one or another. So in fact, we strongly believe at Thermo Fisher that those are two complementary technological approaches that can help each other eventually to get the best possible answer to make the most informed next clinical decision.
Fantastic. So opening up and kind of building off of that, so we have these two genomic technologies which are really gaining steam in the community overall. We're looking now to the current standard of care tests that happen for different cancer types. So knowing that digital PCR and NGS are really gaining in popularity, to all of the panelists here, do you envision a time where we might be able to lengthen the period of time between routine scans or the more traditional orthogonal tests that are performed during a cancer patient's journey? And do you feel like these technologies through the avenue of liquid biopsy could eventually replace these tests or act more as a companion tool? And so I'll open this up to whomever would like to kick off the conversation there.
I can give you my thoughts a little bit on this. Will it replace any sort of radiographic measurements? The answer is no. I mean, we still have to look at the tumor, and we have years and decades of understanding how to measure tumors and how to manage care based on RECIST 1.1. Our clinical trials are based on RECIST 1.1. Our clinical management of patients are based on RECIST 1.1. The field, I think, is very early, but it's promising, where we have demonstrated that circulating tumor DNA or the kinetics of ctDNA is related to biological function. So the dynamic study, for example, showed that post-surgery, you had a rapid decrease in ctDNA, and the presence of ctDNA after post-surgery predicts a not favorable outcome. We also demonstrate that in the EGFR field, as well as melanoma with BRAF 600, that if you have targeted treatment, that you see a rapid decrease in the mutation of interest, and that clearance of that is actually predictive of a favorable outcome. So we have those pieces of evidence. We have new evidences demonstrating that with detection of minimal residual disease can be used to at least change clinical trial, or at least make clinical decisions that actually we monitor that patients. But I do think that as long as those tests are not cheaper and not faster and not scalable, and I mean, we can think about how scalable will look like, that imaging between 6 and 8 weeks will remain the norm for the foreseeable future, because I think we still have not demonstrated prospectively that those monitoring really will make changes in outcomes. So I think we need to be realistic. I like what Dr. Lusa said, that it's a very promising and very revolutionizing field, but I also think we need to think about what clinical practice is and what it takes to demonstrate clinical utility. It's easy to demonstrate clinical validity, that the assay works valid, but we need to demonstrate that the utility of the test actually improve patient outcome. So those are my thoughts, and so it's promising, and we should do more of the tests. But to change radiographic measurements, I think a couple years behind them.
Sure. So to kind of summarize, that you feel like one of the major challenges that has to be overcome for both of these genomic technologies to become more commonplace, or even kind of more predominant than some of the current orthogonal tests, is really this demonstration of not just clinical use, but clinical utility. So actually being able to make diagnostic decisions based on that. So we need more of that research.
Exactly. So I'm just going to sort of clarify. So in the EGFR space, and Dr. Kao knows this, you can detect the driver mutations. You can treat with a third generation EGFR TKI, and you see the decrease to non-detectable levels. As with all targeted agents, you will have recurrence of disease. 2 years, 3 years, 5 years. And studies have shown that ctDNA progression, the median time is about a month before you see a clinical radiograph, but you see also some which are earlier. What we have not demonstrated right now is that we can change treatment based on a ctDNA test that will make a clinical outcome either better or worse. And those studies really need to happen. They need to occur very smartly, and we need to think about studies. And we have some in the immuno-oncology space where we either intensify with chemo based on a ctDNA test. But I think monitoring response and resistance is a very interesting space, but we really need to demonstrate clinical outcome and clinical benefit for the patient.
Absolutely. So then to Dr. Bilbao and Dr. Kao, do you feel that's pretty consistent from your experience for the use and application of these tools for cancer research and management?
Yeah, I agree with what Dr. Pollard just said, and I think the technology is good and technology is here. And so what it needs to be done is to show the clinical benefit using this technology, and there is still a lot to be done. Like, we are very interested, and he just mentioned, if you detect the early recurrence before imaging can detect, and if we give patient preemptive treatment, will there be any clinical benefit to patients? And that is a question we would very much like to learn. So what I'm excited is about the digital PCR. It can be more precise and accurate and quantitative. Then we can have the right tool to do the clinical study and to show how it can be used for the patient's benefit. One of the things is, as you mentioned in your question, is whether the use of the liquid biopsy digital PCR can lengthen the period. And we are using the imaging studies to detect recurrence. And I do believe we can use the more sensitive digital PCR, and probably we can reduce the interval needed to follow up patients. So I believe certainly the technology would reduce the number of more expensive imaging studies needed to follow the patients.
Probably, if I may add, and again, I echoes the sentiment in here, is that one of the major limitations that has reduced the uptake, especially in the clinical space for liquid biopsy, as we heard, is the fact that most of the performed clinical trials are actually only observational. So what we truly need is interventional trials with the aim, as it was already said, to either make a change in the treatment or to effectively measure that there's a difference in outcome for the patient. That is only something that it's starting to happen now. We will have readout of some of these trials in a couple of years down the road. But in fact, from the clinical standpoint, this has been the major limitation. As we also heard, instead, and again, we take proud of this Thermo Fisher, the technology is very solid. And no matter what we're talking about, NGS or digital PCR, we today are able to offer solutions that can truly enable our collaborators and customers to get the answers that they want. Now is about to put the technology in the right time and in the right space, as again, we heard before, from a clinical standpoint. And that is really something that you can do when you have interventional clinical trials, which is really something that we are as well supporting in partnership with some of our pharma partners.
Absolutely. And so kind of building off this discussion, because we're talking a lot about how digital PCR and NGS are really complementary technologies. So I'll start this question. I'll start off with Dr. Bilbao. Really understanding where the practical synergies are between digital PCR and NGS, and when and where you're choosing to apply each technology to your current research and to liquid biopsy overall for the panelists. So I'll open up with Dr. Bilbao. Where do you really see those practical synergies personally?
As Dr. Lucas said before, I believe that it depends. Okay, so I think that in terms of getting an idea of what you're looking for in a specific pathology, for instance, in an acute myeloid leukemia, I would use NGS as a first step to get an idea of what is really altered in this specific sample, right? But then to follow up these alterations, digital PCR, because of the sensitivity and because also it's more economic technique to follow up, okay? Because really the assays are not expensive at all. And then on the other hand, if you are looking for a specific alteration which is present in a specific, for instance, lymphoma, in the case of Waldenström microglobulinemia, for instance, that you're looking for a specific mutation, and what you receive is, for instance, not the tumor sample, but blood sample, but because many times we in lymphomas, what we get is bone marrow or peripheral blood, which may not be infiltrated by the tumor. So we need sensitivity. And I would use digital PCR to look for this specific mutation in the gene that causes this disease, which is the MYD88 gene. So it depends.
Great, and I'll open that question up to all of our panelists just to get your thoughts as we kind of wrap up this discussion on the practical synergies between digital PCR and NGS, and really where you find them landing in each of your research areas.
Well, I like to maybe like pose a rhetorical, but also a question to everybody. Where the technology lands, I think is also really dependent on the geography of the world. And we have here experts from Taiwan, and I have an interest in Southeast Asia, tests in Southeast Asia. So if reimbursement is difficult, and if you have a large population that has to choose between expensive tests and cheap tests, I think we can be very smart in developing tests by digital PCR that cover most of the genes of interest. So let's say you have a diagnosis of lung cancer. You can generate a digital PCR test which is more cost effective, covering KRAS, EGFR, RET, ROS, BRAF. So you can cover the majority of mutations quite cheaply. Reimbursement is obviously an issue in the first country, but the MOI test that actually was developed in China is actually a very good test that actually works. You can think about a digital PCR kind test that actually would help this. So I think it's not as simple where I'm going with this, is where you have, we test with NGS first, and then we test follow up. Like a business idea always I had was like, okay, we offer an NGS service, and then you get 3D PCR tests for monitoring for free. Buy one, get 3 for free, but it's not as easy. It's not even that easy in the United States. Not everybody goes to Dana-Farber Cancer Institute. Not anybody goes to MD Anderson. Most community hospitals don't even do molecular testing for tumor tissue in lung cancer. So I think we need to sort of like really understand where the field is and the geography where we would like to use the tests. Now, from a technical aspect, in a perfect world, if we can do those tests cheaply, fast, and if you can think about some of the logistics around making a liquid biopsy test work from blood draw to actually cfDNA analysis and actually calling ultra rare variants, it would be great to actually give everybody a very quick test by NGS and then follow up by ddPCR, by digital PCR. But I think you also need to understand there's always a reimbursement issue. And it gets more complicated as you go east, meaning Europe is more complicated than the United States, and then you go into Asia. So that's my thought on technology, and we will really need to use it. But I'm really interested what my panel colleagues are thinking about that.
I do believe the NGS and digital PCR are complementary to each other, and they have different purpose. And so the challenge is how to use them to meet our goals. So at our institution, our approach is try to use NGS technology to detect mutation in biopsy or resected tumor tissues. We then selected the appropriate mutation for sensitive and quantitative digital PCR to monitor treatment response and to detect early disease recurrence and emergence of drug-resistant mutations. But I think one important aspect is to reduce the technological complexity and make it more fully automated. And I think it can be done. And nowadays, because it's in Taiwan, there is a regulatory change, and they make the laboratory development test is extremely difficult to get reimbursed, and they only go for IVD. And as a result of that, we found out in order to get the IVD, we found out there are very good fully automated instrument. Then you can cut the formalin fixed tissue sections, just throw it into the cartridge, and they tell you what kind of lung cancer mutation you have. So that, and I believe digital PCR can be done too. All you need to do is put in the plasma, and then you can have the result coming out from the other end. And so when we can do that, and I think, and then combine with the progress in clinical trial, clinical study on this technology, and I believe one day two technologies can be very useful and to improve the cancer patient outcomes. And I'm very hopeful, and especially I believe Paul should be able to do it to fully automate the digital PCR. All we need to do is a minimally trained individual, put the plasma into the well, and then out with the result from the other end.
Well, thank you, Dr. Ka, for giving me a very high ambition to to accomplish. But I think from the panelists, we can clearly see that the trend is about bring the technology easy to use, make it less complex, and then more affordable, and then that will facilitate actually the clinical trials or the studies we want to do. If you think about studies that can take years or even take many patient samples, this processes, like Dr. Cloud mentioned earlier, every step is just complexity. You can add noise to your answer. So if the trend sounds like we would take it by heart, is that we want to move the technology, make it even simpler, easier, and more affordable. And then we can support more of the cancer researchers to uncover the secret behind the biology, and then use that to improve the patient outcome. I believe the clinical relevancy and actionability definitely sounds like that's the area we need to focus on. But behind that, it has to be technology that's easy to use, and then make sure we're not complexity, making the clinical trials or researchers' life more complicated.
And perhaps if I can also add on that, what we also need to do, and that's exactly what Thermo Fisher is committed to do, is to make the two technology interchangeable in the way that you can switch from NGS to digital PCR and back to digital PCR and NGS, and combine the result together within a unique bioinformatics solution that allows to track and monitor, for example, the evolution of a patient when you are doing MRD testing. That is really something that is going to be critical to enable, along as Paul said, with the easiness of use and the ability to fully automatize the system. That is what really will enable to have a true end-to-end solution. No matter what you're using, depending on the different clinical stage and situation of the patient, you can seamlessly switch from one technology to another and still combine the result that they will, represent a unique record. Almost you're doing a snapshot during, different time of the patient disease, and you can still track all of that together without even, having any kind of issue. So I think that is really something that we are committed and willing to work on, to truly democratize the access to those technologies in as many places as possible in the world.
All right, I think that's a fantastic point to start to wrap up our discussion on. So I'll invite our panelists if there's any final closing thoughts. We're happy to hear them now. And otherwise, I will thank all of our panelists for participating in this webinar event. It has been an incredibly rich discussion on the state of liquid biopsy research, as well as the state of digital PCR and NGS technologies and how they're really being used now and how we feel they're going to impact clinical cancer research applications both now and in the future. Additionally, I'd like to thank all of our attendees for participating in this panel discussion. We've been taking all of your questions submitted during the event, so please stay tuned for an after-event summary. And thank you so much.
Thank you, everyone.
Thank you.
Thank you.
Thank you.